Thanks a lot, Dr. Rizwan, for a very insightful talk. Just to add like few things, uh, uh, almost like half of our group is targeting healthcare research. I mean, ranging from imaging and sensing. And recently we have inducted majority of them biomedical engineers
Oh,
just
that's
to
wonderful. That's good to know.
yes, it's like most of them from UET, you see, normal, they have specialized biomedical engineering programs. So several people actually joined from that, and we want to further enhance our capacity building in this particular domain. And that's why we wanted to have you to see what could be the possibilities on our side, because biomedical engineering is like a broader term. Uh,
Yeah.
and as of now, our focus obviously remained on the imaging and sensing side, optical imaging and sensing mostly. And this talk actually gives lots of interesting ideas in terms of biomaterials and obviously how we can integrate AI and scientific computing into this direction where that could be thought of perhaps our possible contribution or role on this whole kind of direction. With this, I will allow the audience if they have some questions. So please go ahead. And then maybe I think I will have a couple of questions and then detailed discussion because I have taken the several notes. So maybe later on off channel
Sure.
we can have some
Absolutely
discussion.
sure.
Yes, thanks. So students, teachers, staff, everybody, if they have some questions, please go ahead.
Sir, I would like to ask a question.
Yes, Usman, please go ahead.
Yes.
Assalamu alaikum, Dr. Rizwan. Myself, Usman Ali Shams, and I have done my master's in hematology from University of Health Sciences, Lahore. I've done a couple of research work on the leukemia side, actually, cancer side. So also we have tissue culture techniques in our lab at the Department of Hematology. So, if is there any steps you have taken so far by undergoing all these research work you have done so far in your students? Is there a special SOPs for the tissue culture while the process? And the second question is for the liver bile duct production. As far as I know, that cross matching is mandatory for a liver transplant. So, what have your team or your research have done for the cross match kind of like mismatch not happens for the regeneration of liver?
Yeah, so to answer your first question on the tissue culture, so obviously as as this presentation shows, my background or my roots may be in or in material engineering and metallurgy, actually from Punjab University. But the current focus of the lab, as you can see from this presentation, is highly multidisciplinary. We are much more of a biological lab as well, as much as we are a biomaterial lab. So we use a range of cells starting from basically tissue-derived cells to cell lines purchased from commercial sources to stem cells that are differentiated in the lab. So we have obviously a dedicated tissue culture room with different hoods and all. So and the protocol really depends on each cell type, right? Whether it's a cornea cell derived from human cornea tissue or liver cancer cell line or Hep G2 cells. The protocols obviously will depend on what kind of cells we are using, and similarly, the precautions or the amount of care required for those cells as we are culturing them also depends. For example, if we are culturing iPSCs or induced pluripotent stem cells versus a regular cancer cell line, the considerations are very different. So yeah, there are protocols in summary for tissue culture. To answer your second question, I'm not sure if I understood correctly, but I guess you're talking about the transplantation of the liver and its functions. So the current focus in the lab is creating an autologous tissue from patients, let's say a skin tissue or a blood tissue, converting those cells to induced pluripotent stem cells, and then converting induced pluripotent stem cells to liver cells of different types. As you know, or as you might know, liver has different types of cells, including cholangiocytes, which are bile duct cells, hepatocytes, which are almost 70 to 80% of the liver tissue. It also has Kupffer cells, stellate cells, right? So we want to create all these different cell types in the liver, and then we want to integrate them together to form this basically these micro tissues and assemble these micro tissues into a bigger structure. The goal of using tissue or starting this with the patient sample is so that we don't have immune issues, right? When we are doing the transplantation, because we start with the patient sample itself, then if the tissue is created for the patient itself, the patient does not need to take any immunosuppressive drugs for the rest of their life. That is the goal. Now, how long it will take, we'll see. But it's pretty exciting time. We want to create a fully immune competent tissue that is derived from the patient's own cells instead of basically the cells from some other patient. Yeah.
Thank you, Dr. Rizwan, for the explanation. Thanks so much.
Thanks. So maybe we can.
I have a question.
Yes, please. Yamal, I think
So
you
I was
introduce
thinking,
briefly first, once every time, and then go ahead.
So I was thinking that we use some other thing with the hydrogel, and hydrogel is for transport. But is it possible that we just use the hydrogel and modify it in some way, or it has some structure that influences cell growth for tissue repair, like for healing?
Yeah, it's absolutely. It's actually a very nice question. So first, it depends on the challenge that we're trying to solve. For cornea, it will not work because these corneal cells, or let me go to that schematic. For example, these corneal endothelial cells, they do not regrow. So people have tried a lot of things, but it's very difficult to regrow these cells if they are gone already. So we have to transplant cells. But in some cases, for example, in the case of stroke here, right? So in this case, potentially it's possible to use, and people have been using just the hydrogel without the cells with some growth factors or some other drugs that can be released in the damaged environment. Because we know that in the brain there are progenitor cells or stem cells that hopefully we can control. It's sort of, there is a region of the brain that is dead after stroke, and now we put a hydrogel there, and if hydrogel can release some growth factors or some cytokines to sort of attract or create some gradients that will attract the stem cells into that region, and then hopefully those stem cells can differentiate into neurons or other the brain cell types. So there is definitely potential of just using hydrogels. For example, in wound healing applications or skin wound healing, a lot of the matrices, and I know in IRCBM people have been doing this, that they create these matrices for wound healing applications. We don't necessarily have to transplant cells for every tissue regeneration. It really depends on which tissue we are talking about and how much damage has been done.
Right. Thank you. That was exactly my question. Thank you.
Is there any other question?
Assalamu alaikum, sir. Sir, myself,
Well,
Dr. From Southwestern Medical College, and I'm a PhD scholar. So I just want to ask a question regarding macular regeneration, if there is some parallel studies for macular regeneration for the patients of diabetic retinopathy and hypertensive retinopathy. Are there any parallel studies or some research work under your supervision for, inshallah, in future perspective for the cure or any suggestive leads for that, so that patients can gain, because the diabetic retinopathy is a very, very
it's very difficult. Yeah, it's very difficult to treat. Yeah.
Any biomaterials or engineering techniques so that we can actually reduce that factor for those patients who are actually after the cataract surgery or the macular degeneration, the patient has on a debilitating eyesight weakness and they can lose their eyesight and vision. So
I fully agree. So yes, not in my lab. What I have been, as I mentioned, I have a joint appointment in ophthalmology, and department in ophthalmology folks, they are obviously looking at all different aspects of ocular regeneration and ocular diseases, including diabetic retinopathy. What we have been doing in my lab is not the diabetic retinopathy, but we are focusing on glaucoma. And because eventually what happens with glaucoma, that the increased pressure or intraocular pressure eventually causes the degeneration or loss of retinal ganglion cells, and which are also very difficult. I mean, they don't regenerate simply. So the only way literally is to kind of provide or inject cells, sort of cell therapy, or maybe some gene therapy application where we can inject some genes to be able to sort of improve the regeneration of those cells, or at least hinder or stop the degeneration of the cells because of the glaucoma. So in my lab, actually, one of my PhD student is working on with a collaborator from the ophthalmology, who's basically well known in the field of retinal ganglion cell mechanisms and their regeneration. So we are creating. I don't want to give too much away because this will be posted also on the YouTube. But yeah, we are developing some biomaterials to be able to transplant the retinal ganglion cells derived from iPSCs in an optic nerve crush model.
Okay, sir, it will be a very great thing if this continues to work for those patients, especially of hypertensive, that is hypertensive retinopathy, and further, inshallah, in diabetic retinopathy. Thank you so much, sir, for
I agree.
Thanks, Dr. Humaira. Is there some other question? So, Dr. Rizwan, can you take like 1-2 more questions?
Sure.
Thanks for giving. I think people are excited to see your talk. So
that's good. That's good.
Any other question? Oh, okay. Just a quick comment. What I would like to ask, like you mentioned this fabricated kind of nano microstructures, how they can enhance. So basically, this enhancement is for the case of sensing purpose, right? Sensing and kind of.
Yeah. So in this case, for example, this started with, actually when I was doing PhD in Singapore, the lab was focused on cell surface interactions. So simply put, if cells are going on a flat surface versus if the cells are growing on a structured surface, can they identify, or can they know, or are they conscious that the surface has some structures, and do these structures change the way cells grow? So that was the theme of the lab. And I did identify that indeed cells are able to, and when I say sense, it's different kind of sensing, not the biosensing that you would mostly think of. Sensing as in just being aware that there are patterns there, and those patterns affect the protein expression in the cells. And cells are basically are all about like their gene expression, right? So if we can modify the protein expression in the cells by simply changing the type of structures that are present on the surface, it means we can control the cell behavior, right? So we were using these patterns. We were looking at different types of patterns so that we can identify those patterns that will give us cells that express more Z01 protein. And that was the whole goal, basically, for this project. So we showed that actually some sort of patterns, these 1 micron pillar patterns, particularly with around 9 micrometer gap, they were performing better than other kind of patterns in improving the expression of this protein in these cells.
Interesting. So are you still like looking into this kind of, I would say, nanostructured kind of interactions for your
yes, it's part of actually one of the funded NIH project that we have, that now instead of looking at this, what we are, because cells also secrete different proteins in their microenvironment, and we want to see that can we tune the secretome of the cells, or the proteins that they secrete, can we find or identify patterns that can improve the secretion of proteins? And that is actually very sort of important. Let me explain why that is super important. Because when we want to do this, right, when we want to grow cells on a hydrogel, transplant into the cornea, and then develop or sort of degrade or dissolve that hydrogel film, the rest of it is just a layer of cells now, right? So we think that if the cells can secrete more proteins, that monolayer or sheet of cells will be more stable, because now they have sort of made their own substrate, right? That their own layer of proteins on which they are residing instead of on a hydrogel film. So that's what we are trying to improve the secretion of proteins.
Very interesting. And by the way, for this kind of, you see, nanostructures and also for your
Yep.
biomaterials engineering, so is it like purely experimental based, or there is some part of your group which is focusing on the computational side of these things as well?
Yeah, so people have been trying to integrate the theoretical approaches or like trying to model processes, but there are limits. Because cells is very complex. The processes going on in the cells, it is really, really difficult to model those processes or to predict those processes. For example, I mean, if it's doable, it will be wonderful to just change the parameters of the patterns on the surface and predict something about which patterns will be able to improve the secretion of proteins. That is a very hard problem to solve. I mean, with all the computing power we have, Google has been recently been able to, or the companies have been able to sort of model how proteins fold, and with different kind of amino acid sequences, how their folding or secondary or tertiary structures will look like by using these programs such as AlphaFold. And that we are talking about one protein, right? And in the cells, we have a ton of different proteins, and so many interactions are going on, and so many signaling pathways are going on. So it's very difficult to predict the cell responses unless we have a lot of data. For example, I have a colleague who has done nice work on culturing cells on different systematically sort of tunable patterns, looking at protein expression, looking at cell shape changes, and then doing machine learning to tell that if we have this kind of pattern and if the cells assume this kind of shape. And when I say shape, I meant like polygonal shape, elongation of the cell, or circularity of the cell. And what she showed that we call that morphology or cell shape. So she showed that the cell shape can be linked with the differentiation of the cell. Basically, what she showed that with protein patterns, not protein, the structure relationship with the cell shape and cell shape relationship with the proteins that the cells will express. But it was based on a lot of the experimental data that was initially conducted. It was not purely a theoretical study. But there is some work that can definitely be done. But it will definitely start with the wet lab work.
Great. Thanks a lot. Just like one small last question.
Yeah.
This kind of real-time growth monitoring, is it how it is actually done? Is it like
it is
by normal
that's the
naked
thing.
eye?
I'll go there. So where I mentioned, for example, here, this is where I mentioned, right? I recently saw a paper, I'll be happy to share. They were looking at inflammation, right? So bigger picture, if we're talking about bigger picture in regeneration. So there's a tissue damage, and it happens because of inflammation. Like if there is a stroke, there will be inflammation that will cause degradation of the tissue. So that inflammation eventually goes down. But that inflammation, there are markers, right? There are markers that we know that these markers will indicate inflammation. And those markers are basically proteins, right? The expression of those proteins. So in the brain, if those proteins which are known to basically indicate inflammation, can we measure the expression of those proteins in real time? Or like at different time points after the stroke or after the implantation of the biomaterial, if there is an inflammation going on. So currently, it is actually very difficult to do any real-time sensing. Like one way people can maybe do is if there is a, so if they can engineer cells to have some sort of fluorescent reporter, and therefore if whenever they see a signal, they can image it using from the outside. If the fluorescent signal goes up or down, that will indicate. But it also it depends on engineering the cell first, including that reporter in the cell. So it's purely kind of molecular biology, cell biology kind of approaches that are being used. People have used some sort of, I would say, sensors. Like if we can put something in the brain, let's say, if you're talking about brain, we can take any tissue. Like we can take a liver, right? If you're developing a liver, we would want to know that we have developed a liver. Okay, we bioprinted something, it has cells, it has biomaterials, and cells are producing albumin and secreting some enzyme, just like liver does. But after we put that in the mice, is it still functioning? Like we can maybe measure a few things using blood sample from that mice, right? So we can take blood sample, we can analyze some markers, but it'll be nice to actually have better methods of basically looking at real time. So there are approaches people have done. There are a lot of papers, but it's very task specific or question oriented, that what do we want to analyze? For example, I'll be very interested to analyze inflammation, like how that can be done. Maybe putting some chip in the hydrogel inside the mice and seeing if something can be observed from that, or maybe taking blood sample and then looking for a particular marker, and maybe the methods that you guys develop, if that can provide some signal. So it has to be pretty marker sensitive or marker specific approach of sensing.
Thanks a lot, Dr. Rizwan. So is there some
Hello, sir.
other...
I have a question.
Yes.
First of all, I want to appreciate the efforts you made for this presentation. My question is actually related to the tissue regeneration. In my bachelor's work, I have done work on samarium doped hydroxyapatite that is used for the regeneration of tissues for the bones, right? So over here, you use hydrogel for the tissue engineering for the eyes and
Yep.
brain, right? So I want to ask that the main important thing is the biocompatibility of the tissues and the sample that should be matched. So is there any way that the samples can be used if we can make them biocompatible? The same sample can be used for different parts of the bodies if they are biocompatible, or in the future if they can be used in order to reduce the cost as well as the material variety. Thank you.
Biocompatibility is sort of the very first thing that we would check, right? If we develop any type of hydrogels, like I skipped a lot of material, for example, if I go to this slide and we develop a fucoidin, we cross-link it using this imine cross-link or hydrazone cross-linking or imine cross-linking, then naturally the next question is that, okay, we develop this hydrogel, is it cytotoxic? Like, will the cells grow on this hydrogel? So obviously we would check for biocompatibility, and generally I would say that if we can test excuse me, we can test the biocompatibility in vitro by culturing cells on these materials, or we can implant this material subcutaneously, for example, in mice tissue, and looking at the biocompatibility there. In general, the cells, I mean, if the material is biocompatible in one tissue, it should be biocompatible in another tissue as well. So I don't really see, but obviously we have to check the biocompatibility. Like I'm not sure if I understood your question appropriately, or if this answers your question.
Oh, sure, sir. Thank you.
With this, I think we should thank Dr. Rizwan for giving you see this all this time and very
Oh,
nice
thank you so
discussion,
much for
very
the
thoughtful.
invitation. I'm glad what your efforts and what you're doing for this.
Yes. Thanks.
Thank you so
Thanks
much.
again, Dr. Rizwan. So with this, we will call off the session. So thanks everybody. Thanks for coming and joining. Thank you so much.